I want to talk about one of the most elegant and important systems your body has for preventing cancer, what we might call the molecular brakes that stop cells from growing out of control. Understanding how these brakes work and how UV radiation can disable them really illuminates why some people develop cancer while others don't, despite similar exposure. The most important brake in this system is a protein called P53, and I mentioned it briefly earlier, but it deserves a deeper look. P53 is sometimes called the guardian of the genome, and that's not an exaggeration. This protein constantly monitors cells for signs of stress or damage. When it detects problems, DNA damage, abnormal growth signals, insufficient nutrients, or other cellular stress, it can activate one of several responses. The first response is to pause the cell cycle. P53 can essentially hit the pause button on cell division, giving repair systems time to fix whatever is wrong. If the damage gets repaired successfully, P53 releases the brake and the cell continues normally. The second response, if damage is too severe to repair, is to trigger apoptosis, programmed cell death. The cell essentially self-destructs rather than risk passing damaged DNA to daughter cells. This is a feature, not a bug. It's your body choosing to eliminate one potentially dangerous cell rather than risk that cell becoming cancerous. Now here's the critical part. P53 is one of the most frequently mutated genes in human cancers. In skin cancers specifically, particularly squamous cell carcinomas, P53 mutations are found in the vast majority of tumors. What UV radiation does is cause specific types of mutations in the P53 gene itself. When the guardian protein is disabled, cells lose one of their most important safety mechanisms. They can accumulate additional damage without triggering repair or death responses. The brakes are off. But P53 isn't the only brake in the system. There are other tumor suppressor genes that work in complementary ways. PTN, for example, negatively regulates a growth pathway, essentially limiting how strongly cells can respond to growth signals. The RB protein controls entry into cell division, serving as a checkpoint that must be passed before cells can replicate. BRCA proteins, famous from breast cancer research, are actually involved in DNA repair and maintaining genomic stability in all tissues, including skin. What makes UV-induced skin cancer particularly problematic is that UV radiation can damage multiple brake systems simultaneously. You're not just knocking out one safety mechanism, you're potentially disabling several. And because these systems have some redundancy built in, one brake can partially compensate if another fails. You need multiple systems to fail before cancer really develops. UV's ability to cause mutations across multiple tumor suppressor genes is what makes it such a potent carcinogen. Here's where we can find a more optimistic angle. The multi-hit nature of cancer development means your body has multiple opportunities to catch problems before they become cancer. Even if P53 is mutated in some cells, other protective systems may still be functional and can eliminate those cells. Even if several brake systems are compromised, the immune system provides yet another layer of defense. This is why the vast majority of cells with UV damage never become cancerous. The redundant safety systems usually work. Moreover, understanding these brake systems has revolutionized cancer treatment. Many new cancer therapies work by restoring function to brake systems or by exploiting weaknesses that arise when these brakes are disabled. For instance, some treatments can reactivate P53 function or target pathways that cancer cells depend on more heavily once they've lost P53. From a prevention standpoint, the message is clear. Every time you protect yourself from UV exposure, you're protecting these brake systems. You're preventing the mutations that could disable your cell's ability to regulate their own growth. These molecular brakes are remarkably effective when they're intact. The goal is to keep them that way.